All right, everybody. Hope you're doing well today. Welcome to a Fix Your Gut live stream. I'm your host, John Brisson. We've been making updates to the Fix Your Gut website. Here's where you can download a copy of the Optimate Health Blueprint, optimize your supplement stack, and your gut health too as well. We're also in a partnership with Formline Performance for the liposomal glutathione. This is the old recipe for the old bulletproof glutathione, still some of the best stuff in the market. Of course, we also recommend Smidge, which used to be Gut Pro, as a probiotic that mostly everybody could take. There's no histamine-producing strains or no D-lactate-producing strains. Of course, you can purchase a copy of Fix Your Gut. The blog is now titled as Research. It's moved here. I got some blogs coming out. Jason Hooper's already written some really great blogs. And of course, coaching page offering health coaching, people with a wide variety of issues. And Jason Hooper is offering coaching for a wide variety of issues, diet, exercise. Hooper's way more qualified to discuss exercise regimens and athletic performance. He's the best. He's been helping me out with my exercise routines, and I'm getting in pretty fairly good, decent physical shape. Hopefully I'll be in the best physical shape of my life by the summer. I've been following a lot of Jason's advice, so he's excellent with his knowledge when it comes to athletic performance. So definitely book a consult with Jason if you're having any issues with your diet, or you're interested in biohacking, which Jason is less conservative than I am. His suggestions of biohacking, which, for some people that could be very beneficial. It's not always the best route to be conservative or look at things skeptically as much as I do. But yes, either, get coaching with Jason or from me. So let's begin with today. Here's a video. The study is Toothbrush Microbiomes Feature, a meeting ground for human oral and environmental microbiota. So let's play this video to get an idea of what it's going to be about. I'll read it. A recent study examined toothbrushes to understand how microbes from our bodies interact with our environment. Toothbrushes are a unique component of the built environment that comes into daily contact with our bodies and water. Researchers characterized the metagenomes of toothbrushes from 34 individuals and found that toothbrush microbiomes resemble microbial communities in the mouth. Toothbrush-associated microbes were less diverse than those in people's mouths, but they had broader antimicrobial resistant gene profiles. And microbial community makeup correlated with factors linked to personal health, dental hygiene, and bathroom features. Although this small pilot study must be expanded to confirm the findings, the results suggest that microbes on the surfaces we touch are affected by our health and behavior, and that understanding how we can influence these microbes will help us make better decisions about cleaning and maintenance to promote a healthy environment. Okay, so let's read the background. Interactions between the human microbiome and surrounding environmental influence microbial diversity central to our health and well-being. Microorganisms are transferred to and from hosts through direct contact with surfaces, as well as passive transmission, shedding of particles and aerosols. Remember, you have a microbiome, like cloud around you that you're secreting from speaking and breathing and just off your skin from the air and the wind. So you're shedding your own, microbiome out, kind of like a cloud emanating from you. Accordingly, microbial assemblages that emerge in indoor spaces reflect human activity. Putative selective pressures within the built environment, desiccation, limited resource availability, chemical residues impact potential microbial adaptations and community dynamics. Since the human-associated microbiota in such indoor microbiome studies disproportionately involves the fraction derived from skin, i.e. microbial communities on surface doorknobs, sinks, dust, the question of what happens to those from other body sites, the oral microbiome, when they find themselves in a hostile non-host environment is largely unexplored. Toothbrushes are a unique component of the built environment that comes in contact with our bodies, mouth and hands, and water on a daily basis. Microenvironments on toothbrushes exhibit periodic wetting, variations in temperature, and differences in physical surfaces that may impact the ability of microbiota to colonize. Used toothbrushes are known to harbor a combination of putatively human-derived and environmental microorganisms such as members of Streptococcus, Staphylococcus, Pseudomonas, Porphyromonas, Paravomonas, Lactobacillus, Klebsiella, Fusiobacteria, Escherichia, and Enterococcus. However, since these assessments were largely limited to culture-dependent monitoring for contamination, much remains unknown about microbial community structure and function on toothbrushes. Mixed microbial communities that emerge on toothbrushes may be shaped by a variety of factors, particularly those that influence potential source microbiomes. For example, the human oral microbiome is associated with age, dental hygiene, and clinical health, dental caries, periodontal disease, and even oral cancer. As toothbrushes appear as a reservoir for both human-associated and environmental taxa, their microbial diversity may also associate with features linked to other microbiomes of the built environment, such as how dust microbiota are influenced by indoor chemistry and building design, antimicrobials used in cleaning products or presence of windows, for example. Aside from the limited understanding that antimicrobials is incorporated into oral care products, chlorhexidine, gluconate, and mouthwash nanoparticles on toothbrushes influence bacterial counts on toothbrushes. Factors that more broadly impact the toothbrush microbiome are not well understood. Here we characterize the microbial diversity of 34 toothbrushes collected from randomly matched volunteers. Metagenomes of each sample were compared to those with Human Microbiome Project and relevant indoor microbiome studies, microbiomes associated with dust, sink tap water, and shower heads, to cover attributions to microbiota assembly and characterize potential emergence of antimicrobial resistant genes on toothbrushes. By evaluating metadata on dental hygiene and other personal information provided by participants, we tested the hypothesis that a combination of abiotic and biotic factors is important for shaping microbial communities and resistomes on toothbrushes. So here they, tested, the bacteria concentrations from different surfaces like toothbrush or building dust or tap water or one shower head or the skin or vaginal samples or gut samples and oral samples of the people who participated in the study. And, largely it was found that the toothbrushes mostly matched the oral microbiome. There was a little bit of transfer from the shower head, a little bit of transfer from the tap water, and the rest were unknown. But few was actually, from their gut, their skin, and if they're female, their vagina. It's very interesting. And there seemed to be things that affected the toothbrush microbiome. There's differences between males and females. There's difference between how many people, how much you toothbrush, the age of your toothbrush, which is very important to replace your toothbrush or your toothbrush head, 2 to 3 months, maybe even 1 to 2 months if all possible. Toothpaste brands, because different toothpastes have different antimicrobial mechanisms in them, or even things that even microbes could feast on, like polyols. Use the floss, use the mouthwash where a person stored their toothbrush, was either in a cabinet or a drawer or sink ledge. There's a nearby toothbrush, if they left their bathroom window open, how long they left their bathroom window open, if they had cavities, any this is a pretty domestic pets. This is a pretty varied study. They did a really good job here. There's a lot of data. So let's lead to discussion. Toothbrushes are an interface for microbiota derived from human hosts in the surrounding environment. Most of the prominent members of the toothbrush microbiome were those that are common in oral microbiome. Several conserved oral associated taxia, such as Actomyces, Corneobacterium, Haemophilus, Neisseria, and Streptococcus. So a lot of infectious bacteria. We got Klebsiella, we got E. coli, we got Streptococcus, we got Staphylococcus, we got Haemophilus, we got Neisseria. Play important roles in biofilm attachment and development of super gingival plaque. So if you ever gone a few days without brushing your teeth and you take your fingernail, hope your hands are clean, and rub it against your tooth, if you get kind of like a white, filmy, paste on your finger, you're not brushing your teeth enough. That's bacterial biofilm that's accumulating on your teeth. And over time, those bacterial biofilm, they're like colonies where all these bacteria congregate and live together and kind of form this extracellular matrix for protection. The bacteria start producing acid, and those acid over time is what actually breaks down your teeth enamel. Mechanisms underlying biofilm formation that may have implications for colonization of toothbrush microenvironments. Although there is broad taxonomic overlap with the oral microbiome, especially with buccal mucosal sites, toothbrushes harbor more heterogeneous and less diverse microbial communities. Interesting. So the toothbrushes had a less diverse microbes that had survived on them than what was found in people's mouths. Trends for wide variation across toothbrush microbiomes are similar to what has been described on dust or surfaces in the built environment, i.e. over sinks for human-derived microorganisms. Indeed, many of the less niche-specific taxia observed on toothbrushes are found throughout indoor spaces, members of Enterobacter, Cochira, Pseudomonas, and Strenotrophomonas. So Enterobacter and Pseudomonas are known opportunistic bacteria. Collectively, toothbrush microbial communities range from a robust subset of oral microbiota to mixed assemblages containing more abundant case-specific or environmental strains. Despite lower taxonomic alpha diversity, toothbrush metagenomics contain more antimicrobial resistant genes than oral counterparts. Okay, well, that's interesting. So the microbes in your toothbrush are more resistant to antimicrobials over time than in most cases, the microorganisms that are living in your mouth. And that might be because very harsh and very strong bacteria that are able to produce, complex biofilms or very hardy are more likely to survive on the toothbrush than survive in your mouth. There's a little bit more competition. You also have the innate immune system within one's mouth who's trying to eliminate these opportunistic bacteria. While the majority of the observed microbial taxia on toothbrushes were common members of the oral microbiota, communities that were primarily composed of putatively non-human derived taxia encoded on average higher counts of toothbrush-enriched ARGs. So you would actually also get antibacteria that aren't from your mouth, that maybe you leave the window of your bathroom open a lot, you don't keep your toothbrush in the medicine cabinet, which is where people should keep their toothbrushes or cover their toothbrushes if they don't have a medicine cabinet. Because every time you're flushing the toilet without putting the seat down, you're aerosolizing a lot of microbes, and it's going to land on your toothbrush. Now, granted, this study shows it's not as much as once thought, because it seemed to still be the strong concentration of bacteria in one's mouth or microorganisms in one's mouth or what's actually found on their toothbrushes. Nonetheless, you still want to do that. I do it. I keep my toothbrush, I dry it off after every use, and I keep it in a cup behind my medicine cabinet. Metagenome assembly validated that most of the toothbrush-associated taxa carrying such ARGs, among others associated with microbial communities, dominated by non-niche specific microorganisms. Potential ARG-carrying colonists may have been incorporated from the bathroom sink and local surroundings. For example, toothbrush-associated OQXB, which may confer multi-drug resistance from antibiotics Nutrofuran, tetracycline, or quinolone, is common in tap water resistomes. Yep, that's why I used filtered water. I don't use tap water or bottled water when I brush my teeth. Stressors in the built environment, e.g. temperature variations, exposure to anthropogenic chemicals, periodic dissociation, may play important roles in elevating toothbrush resistomes, and at least for key members that emerge within the microbial community from the environment. For example, triclosan and other anthropogenic chemicals in personal care products, e.g. soap, shampoo, and lotions, that have been widespread in indoor spaces have been linked to multi-drug resistance and dust-associated microbial communities. Yep, that's why I don't use triclosan. Triclosan also is an endocrine disruptor. It's a very hazardous chemical and interferes with thyroid function, sex hormone production and utilization as an antimicrobial agent. In my opinion, triclosan should be banned. Toothbrushes may harbor some similar microbial colonists, especially considering ARG enrichments reflected a variety of multi-drug effects genes, along with putative conferred resistance to triclosan, i.e. FABI and FABG. So they're talking about triclosan causing these bacteria to develop genes that make them resistant to antibiotics. In fact, several oral care products even contain anthropogenic antimicrobials. Triclosan in toothpastes, nanoparticles on toothbrushes may accumulate on or leach from bristles. While such compounds are tended to lessen bacterial load to promote oral health, there are considered undesirable consequences such as induced selection of opportunistic oral pathogens, as noted for Provotella or ARGs. So yes, your toothbrush or toothpaste should not contain triclosan or any like nanoparticles if possible. Silver might be okay if it's embedded into the toothbrush, but I wouldn't recommend triclosan. Or even chlorhexidine. I wouldn't recommend that either. While such compounds are intended to lessen bacterial load to promote oral health. Okay, I read that. Moreover, since most toothbrush-enriched ARGs were still present in microbial communities containing primarily human-derived taxia, a bit to a lesser extent than those composed of mostly case-specific or environmental strains, unique toothbrush resistomes may be partially related to a selection of human-derived taxia best suited for survival under harsh-built environmental conditions. Microbial communities on toothbrushes appeared to weakly associate with several factors previously shown to correlate with microbiomes of the oral cavity: gender identity, missing teeth, adenoid tonsil removal, and the ownership of domestic pets. While dental hygiene frequency, toothbrushing, flossing, mouthwash use frequencies was inversely related to the microbial taxonomic diversity on toothbrushes, which is consistent with the earlier report of mouth rinse lowering bacterial counts on toothbrushes. It was positively linked to the resistome. Although plausible that there could be less accumulation of human-derived taxi on toothbrushes of people who more frequently practice comprehensive dental hygiene, so if you have a cleaner mouth, you're going to have less microbes on your toothbrush. Less plaque on teeth leading to less transfer to toothbrushes. Putative source fraction of the toothbrush microbiota do not significantly associate with oral care, so it's not that much of a difference. Thus, the relationship between more frequent dental hygiene and a broadened resistome suggests possible selection among the mixture of human and environmental-derived microbiota. The positive association with the toothbrush resistome may be partially due to more frequent incorporation of ARG-carrying members of oral microbiota or raised surrounding dust microbial community. Links between toothbrush microbiomes and health, at least a potential biomarker, warrant investigation with a larger cohort with a balanced clinical metadata. Relations between toothbrush microbiomes and features of the surrounding environment, window presence, and frequency of opening are consistent with previous work showing building design to weakly influence indoor microbial communities. Although there is a strong desire for using bio-informed design to create healthier indoor microbiomes, the vast majority of observed indoor microbiota are likely dead or metabolically inactive, because they're not close to host. And no so small part due to lack of moisture. However, there are niche and built environments that, like toothbrushes, are frequently wetted, sponges, dishwashers, and washing machines. Toothbrush microbiome is similar to these in that they appear to be hotspots for putative, opportunistic pathogens and antimicrobial resistant, yet different in particular microbial membership. Toothbrushes contain primarily oral-derived taxia. And in biofilm dynamics, since our exploratory analysis was based on correlation and did not control for variables that may drive the microbiome, future studies are needed to better understand how we might be able to target, design, operation, use of different components of the built environment to promote microbial diversity that limits pathogens and antimicrobial resistance on toothbrushes. As the scope of our pilot study was to define the toothbrush microbiome and potential driving factors, limitations include the relatively small sample size, 34 people, and the lack of prepared donor samples. Perhaps a larger cohort would improve effect sizes for the weak, yet potentially important trends uncovered in this pilot study. Microbiome associations with oral hygiene products, missing teeth, adeno tonsil removal, presence of bathroom windows, toothbrush storage location. After all, though indoor microbial communities are impacted largely by the environment, such features often have small or negligible associations with community metrics. Moreover, the comparisons made between toothbrush microbiomes and human-associated and built environmental counterparts from separate studies are quote-unquote high level due to site specificity of microbial strains and possible confounding variables across data sets. Variation in the treatment of actual sink water in subject households versus tap water, reference data sets, batch effects, and sequencing preparation. I'm pretty sure if you use filtered water, you'll have a healthier toothbrush, and a healthier mouth, and a healthier body. For example, some of the putative oral-derived microbiota may have been overestimated and rather had come from other human body sites. While members of the toothbrush microbiota putatively derived from the human microbiome were most common across oral metageomes, hence to human fraction predictions made by Source Tracker. Many of the same microbial species occur in the skin, vaginal, and gut microbiomes as well, e.g. Streptococcus and Veillonella. That's true. Likewise, some of the prominent taxon toothbrushes that contain mostly non-human derived microbiota may still associate with human body sites, e.g. potential enteric strains of Enterobacter Klebsiella. Remember, Klebsiella is a possible cause of alkalizing spondylitis or rheumatoid arthritis. I've coached many people with Klebsiella dysbiosis having one or the other, and once you get that Klebsiella dysbiosis under control, AS or RA goes into remission. Future longitudinal studies monitoring microbiomes of toothbrushes and donors, perhaps incorporating genome-level sequence variant comparisons for tracking, will be essential for making stronger conclusions about community assembly on toothbrushes and potential driving factors. Nevertheless, consider distinctions from the human microbiota that may indicate local selection. Toothbrush microbiomes appear to be an important reservoir for the antimicrobial resistance. It remains to be determined whether this sink may, in turn, become a relevant source of ARG dissemination. Remember, that's antibiotic-resistant genes for microbes. In the human microbiome and indoor environment. So one way you can sanitize your toothbrush is very simple. Just get 3% hydrogen peroxide and put the head of your toothbrush in the hydrogen peroxide and move it around really well and leave it in there for a few minutes. And then take it out and dry off the bristles and put it back, in a cup in your medicine cabinet or a container in a medicine cabinet once it's dry. And, that'll, allow the toothbrush, it'll kill most of the microorganisms on the toothbrush. And it'll be more healthier for you. Another way, of course, is I own and use the Brisson toothbrush. So it has UV light that you can use to kill bacteria both in your own mouth and plaque and bacteria that would be living on the bristles. And you have the red near infrared light, which reduces gum inflammation and helps the teeth to repair. So I highly recommend the Brisson toothbrush. Again, you could take... You can move it around in some hydrogen peroxide. You can't soak it in it, but you could take a little bit of hydrogen peroxide and kind of whip it around the bristle of the toothbrush to clean it further and dry it and keep it in, your medicine cabinet or some sort of container. And then, just turn the blue light on and it'll kill some of the bacteria on the both on the toothbrush and in your mouth. So this is a great toothbrush. I highly recommend it. So let's talk about oral care for a minute. Here's an excellent article I did. 3 oral health mistakes slowly making us ill and what to do instead. So 3 mistakes are mercury amalgams, root canals, and the use of fluoride in toothpaste. So I definitely recommend reading this. I also have a good part of what can be done to improve your oral health care instead of using fluoride. So yes, I do not recommend the use of fluoride for the quote-unquote strengthening of teeth. I don't believe that the so-called benefits outweigh the issues associated with fluoride use. Europe doesn't use fluoride to prevent dental caries. Instead, they use the polyol xylitol, which bacteria have issues fermenting xylitol. Is also able to break down biofilms within the mouth. So yes, definitely if you're wondering why I don't recommend fluoride, this article definitely will break it down. And actually, I'll probably read it someday for another video. But yes, if you're using fluoride toothpaste, I definitely recommend that you talk with your oral health care professional. Hopefully it's a biological dentist. That you should stop using fluoride and instead using some other toothpaste. I'll have toothpaste recommendations in a minute that does not contain fluoride for proper oral health. But yes, fluoride is definitely not good. Not good. Another byproduct in toothpaste is carrageenan, which is a brown seaweed extract. There are some human studies to show that it may cause intestinal inflammation. It may affect the microbiome. There's consumer advocacy groups that are calling for carrageenan to not be used as a thickener. So even though, Chris Kresser says there's not as much information he would like, on the carrageenan and effects on the human microbiome and humans themselves, maybe if triggering gut inflammation or leaky gut. However, he does think caution is warranted, and like me, I have the same conclusion, which is, yeah, there's been a lot of people I've coached who don't feel good ingesting products containing carrageenan. It can cause intestinal inflammation and gastritis, inflammatory bowel disease. It can worsen it. So, yeah, maybe you should just avoid it. It's easy to avoid for the most part. So, yeah, personally, I adhere to the precautionary principle for anything I eat. In other words, in the absence of proven safety, I choose to avoid foods that have questionable adverse effects. Carrageenan fits this description. I agree with you, Cresser. So, a couple of toothpastes that I do recommend. I recommend Earth Paste by Redmont. It's pretty much just bentonite clay, the Redmont salt from Utah. Some essential oils. They do put a little tea tree oil. If you're worried about tea tree oil being a little too broad spectrum into your mouth, and you can use a lemon twist with the kids. It just has D-limonene. There's no tea tree oil. Has some xylitol too as well. So some people are concerned about the bentonite clays containing too much lead. The lead amounts in the bentonite clay are next to none, in my opinion, that they use in Redmont. It's tested. You can go look. You're probably consuming more lead in leafy green vegetables that you're ingesting than actually would be in this earth paste. Besides, you're not even swallowing the earth paste. You're just using it and spitting it out. But it's what I use. If you're worried about the bentonite clay, EcoDent Extra Bright works very, very well as a tooth powder. It doesn't contain any of the negative things that we mentioned earlier. It's just sodium bicarbonate, tartaric acid, which is found in grapes, sodium menthol cocoyl taurate, which is a soap made from coconut, calcium peroxide, dicalcium phosphate, dihydrate, calcium carbonate, which is calcium. This is just phosphate. Magnesium carbonate, sea salt, silica, peppermint oil, anethol, which is I want to say it's a main component of phenol, but I could be wrong. No, it's camphor. See, I was wrong. So it's organic, camphor, guar gum, and myrrh. So pretty good ingredients. I've used it before when I've traveled. It's good stuff. Next, you have Uncle Harry's Remineralizing Tooth Powder. It's another good tooth powder. It does contain some bentonite clay, though, however, but it doesn't contain carrageenan or actually, this one just contains calcium carbonate, mustard seed powder, peppermint, eucalyptus, clove, wintergreen, and oregano. So again, another good tooth powder if you want to use it. Now, if you need a remineralizing toothpaste, you have a lot of cavities, or you've had cavities previously beforehand. Now, granted, the ingredients in most of your remineralizing toothpastes are not the best, but it does contain, nano hydroxypitate calcium, which can help remineralize your teeth. This is one of the best ones I can find. It just has a little bit of sodium benzoate or benzoic acid as a preservative that I'm not necessarily too healthy about. Just have a little bit of methyl sulfur methane, so people with methane or hydrogen sulfide dysbiosis, you definitely want to make sure after you're done, using toothpaste, you should wash your mouth out, which most people should do anyway. But if you have a lot of dental problems, if your teeth are not strong, if your enamel is not strong, then even with a few questionable preservatives that this Ella Mint toothpaste has, I still would definitely recommend it for remineralization compared to some of the other ones that have, sucralose or which is an artificial sweetener, chordated sugar. It's very bad for the microbiome. Or a lot of them have artificial sweeteners, which are very questionable, or fluoride in them. So this one's one of the better ones. Next, oil pulling. If you're just oil pulling, you put a half a tablespoon of coconut oil in your mouth. You swish it and pull it between your teeth for 5 to 10 minutes. You spit it out in the trash can. You don't want to spit it out in your sink because coconut oil can solidify in the pipes. It can block your pipes. And then you wash your mouth out very well and then brush your teeth. Oil pulling is very good about breaking down biofilm and getting food that's lodged in between the teeth out and pulling bacteria out, and so there's less toxin production. Of course, if you have a more infected mouth, you might need to do something like this Dale Audrey oil pulling natural mouthwash, which just contains organic sesame seed oil, sunflower oil, olive oil, clove oil, neem oil, myrrh oil, and oregano oil and pomegranate extract. So if your oral health is a little bit challenged, you got some root canal, some infected teeth, some cavitations, you might want to go more with a stronger oil pulling oil, in my opinion, than just using standard coconut oil. For mouthwash, if you want just a basic mouthwash that's environmental working group verified, I recommend Dr. Bright. Not really anything bad here that I can really say. Aloe, glycerin, coconut oil, xylitol, mint flavoring, tulsi, which is holy basil, neem, vitamin C, some stevia, sea salt, and activated charcoal. So pretty good ingredients. Pretty good. Also, environmental working group verified, so it's not toxic for your mouth or for your family. This is just a good basic mouthwash. If your teeth are healthy, you can use this and maintain really good, health. Might have to be careful with the stevia, though. Some people who are allergic to ragweed, there's ragweed cross-reactivity, oral pollen allergy syndrome with stevia. So if you are allergic to ragweed, you might be allergic to stevia as well. For me, for example, it makes the roof of my mouth itch. If you need a stronger mouthwash, then you can either go with Miracle Mouthwash by Uncle Harry's or the Elemental Silver Nano Silver Mouthwash. This would use colloidal silver. This one uses plant-based nano silver. I know some people have concerns with nano silver and its ability to accumulate in cells. So you can take your pick between which one that you want to do. The silver shouldn't, you use these mouthwashes, you swish for 30 seconds to a minute, you spit them out, and then you swish around with some water and you spit out the water too. I'm not really too concerned of silver accumulating in the body if you're using these. But again, I only recommend these two if you have issues with oral infections, you got an infected tooth, you have cavitations, you just have really bad oral health. You need to heal some things before you get, if your gums are bleeding, if you got some sensitive teeth, if you got some teeth pain, these are something you want to use. You want to use these on a daily basis. If your teeth are very healthy and you very rarely get a cavity and you take very good care of your teeth, you don't want to use these. But if you have some issues, you might need to use these. I mean, the element of silver is pH balanced with the calcium. It's good stuff. I mean, if needed, you should use these. Of course, you have an oral copper tongue scraper that's made out of copper. There's no rubber grips or anything, so you can take this oral copper tongue scraper and scrape the back of your tongue with it and get some of that biofilm out. Most of the time, white tongue is bacterial biofilm or from a dry mouth from reflux. It's rarely from candida unless you have oral thrush, where you have these white raised patches all over your oral cavity. Then yeah, that could be from candida, but most of the time the white that you see on your tongue is either bacterial biofilm, which can be taken off from a copper tongue scraper, or is from having a dry mouth. And finally, dental floss. I recommend this bamboo peppermint infused candela wax. So it's natural wax. It's not plastic that you're putting in your mouth, leaching, bisphenol A or phthalates or whatever that could cause issues with hormone production and disrupt your endocrine system. So yes, some sort of wax like this, if you're going to floss your teeth, would be a good idea for sure over your standard, plastic petrochemical based waxes that most people use for dental floss, which could disrupt the endocrine system with a lot of plasticizers that are being used. I'll have eventually all of these links for all these products in the show notes. All right, that's it, everybody. How clean is your toothbrush or your mouth? Depends on a lot of factors. Hopefully I've given you some good tips of how to keep your toothbrush clean, how to keep your mouth clean. Take care, everybody.